In the modern obesogenic environment, roughly 15% of children are overweight and another 15% obese by kindergarten. Overweight or obese status at this age greatly increases the risk of adult obesity, often leading to an earlier onset and more severe complications that arise from obesity. Obesity is a highly multifactorial phenotype of which the importance of the hypothalamus, a regulator of energy balance, is well established. However, little is known, particularly in humans, about the developmental origins of variation in hypothalamic structure and function. Here, we focus on maternal free fatty acid concentration based on the logic that FFA concentrations are elevated in human pregnancy, that they freely cross the placenta, and that the hypothalamus is a sensor of fatty acids. A more specific example demonstrating the plasticity of hypothalamic development in the context of free fatty acid concentration is shown in a recent study by Sébastien Boré and colleagues. In this rodent study, they replicated the now common paradigm of maternal high-fat diet during pregnancy, eliciting offspring obesity characteristics including changes in adiposity and metabolism. More importantly, they observed a marked reduction in axonal outgrowth from the arcuate nucleus of the developing hypothalamus when exposed to saturated free fatty acids using in vitro explants. Critically, recent evidence suggests that the human hypothalamus can be differentially phenotyped in the context of obesity using diffusion-weighted MR imaging. In this study by Vette et al., they report an externally replicated positive association between BMI and mean diffusivity within the hypothalamus. In the context of the current hypothesis that maternal FFA concentration would blunt axonal outgrowth and thus lower the barrier for the unrestricted diffusion of water, as depicted in the following cartoon, this phenotype would present as a positive association between maternal FFA concentration and mean diffusivity. Combining three key pieces of evidence: 1. The maternal obesity is associated with elevated concentrations of circulating free fatty acids. 2. The maternal free fatty acids can impact the structure of the developing hypothalamus. And 3. That the structure of the developing hypothalamus can be indirectly characterized by diffusion-weighted imaging. We set out to test the following hypothesis. First, maternal saturated free fatty acid concentration during pregnancy is associated with newborn offspring hypothalamus mean diffusivity. And second, newborn offspring hypothalamus mean diffusivity is positively associated with offspring body fat percentage at 5 years age. This overview slide presents the longitudinal study design. Pregnant mothers were recruited in early pregnancy, and free fatty acid concentrations were characterized in early, mid, and late pregnancy. Babies born to these mothers were imaged shortly after birth using diffusion-weighted MRI, and again in early childhood using DEXA to ascertain whole body fat percentage. I will now provide a more in-depth look at each of these three measurements. Maternal saturated free fatty acid concentrations were z-scored and averaged across pregnancy after median replacement for data missing at random. All 14 saturated free fatty acids measured were then averaged together to create a composite measure of one saturated free fatty acid concentration per mother. A 42-direction DWI-weighted sequence was collected in all newborns as previously reported. Image preprocessing was performed in accordance with the neonatal diffusion pipeline recently published by the DHCP group, spatially transformed using a sample-based template, and average hypothalamus mean diffusivity extracted according to a high-resolution probabilistic atlas of the human hypothalamus at a partial volume threshold of 60%. Here you can see the distribution of body fat percentage in early childhood, a roughly age-independent time period that represents a local minima in adiposity during the adiposity rebound. For the purposes of this study, body fat percentages were a priori adjusted for sex and age at DEXA scan. Here we present our primary finding that maternal saturated free fatty acid concentration during pregnancy is positively associated with offspring hypothalamus mean diffusivity. Several considerations strengthen our confidence in this finding. For one, the association was robust to different methods for defining free fatty acid concentration, including PCA reduction across the entire metabolomic profile, and more simple measures like raw concentrations of palmitate, the most abundant fatty acid in the human body. The association was also robust to definition of the hypothalamus ROI across different partial volume thresholds and survived correction for several socio-demographic variables. Further, the association was found to exist above and beyond the effects of maternal BMI and global mean diffusivity, suggesting specificity to FFAs in the hypothalamus. Further, we observed a positive association between newborn mean diffusivity and the hypothalamus and body fat percentage in early childhood. This positive association suggests that the newborn phenotype of hypothalamic mean diffusivity is of functional relevance. In addition, this association survived adjustment for several important potentially confounding factors, including SES and breastfeeding status in early life. In conclusion, we present evidence of an association between maternal free fatty acid concentration during pregnancy and newborn offspring hypothalamus mean diffusivity. The direction of this effect is consistent with animal literature suggesting a reduction in arcuate nucleus outgrowth in the presence of high concentrations of saturated fatty acids. In addition, we demonstrate the functional relevance of this newborn phenotype in the context of a childhood obesity that is consistent with observed associations in human adults. Collectively, these findings support an underlying neural basis of childhood obesity risk programmed, in part, by in utero exposure to variation in maternal nutritional status. The identification of brain-related biomarkers of childhood obesity risk that predate the influence of the postnatal obesogenic environment may contribute to an improved understanding of early identification of at-risk individuals and intervention targets for primary prevention.